afternoon, everyone. I'm Alicia Gonsalves, and this is Arya, Prabhu. We are students of Basabdatta Sen College of Engineering, Pilani Goa. The other co-authors of this paper are Karen Pereira and Fatlabas, also of ICETCI, and Dr. Basabdatta Sen Bhattacharya of BITS Pilani Goa Campus. Our paper is entitled "A Brain-Inspired Neural Network for Image Classification." To begin with, let's go through some of the background that's essential to understand our work. Starting with the brain cell or the neuron. So the neuron has two main parts, dendrites and the axon. Electrical signals are received via the dendrites and transmitted along the axon, and then electrical energy is converted to chemical energy to cross the gap between neurons, which is called the synapse. So these electrical signals which neurons use to communicate are called spikes. Spikes are instantaneous in nature, that is, the time interval during which they occur is negligibly small, which is why they are treated as all or nothing events. And that's why we can represent them in binary, a 1 for the presence of a spike and a 0 for the absence of a spike. So we all know what a neural network is, but what's a spiking neural network? So a spiking neural network is a brain-inspired neural network which uses models of spiking neurons as the nodes. Convolutional neural networks are also brain-inspired, but their activations are continuous or real values. So even minor changes in input can activate a node, unlike in the brain where activations are sparse. So that was our main motivation for researching SNNs, is the computational efficiency and the sparsity. There are many neuron models that can be used as the nodes of SNNs. The most biologically realistic one is the Hodgkin-Huxley model, but this is computationally heavy and complex to implement. The leaky integrate and fire neuron model, on the other hand, is widely used and it balances biological realism with computational efficiency. And that's why we have presented an SNN which uses the LIF model as its nodes. So in the illustration on the left, you can see a stimulus being received via the human eye in the form of light on the retina, and it's being transmitted to the thalamus, and from there the signal goes to the visual cortex, from which it receives feedback. There's a structure called the thalamic reticular nucleus which intercepts these signals being transmitted between the thalamus and the cortex. On the right, you see the three main neuron populations involved: the thalamocortical GA cells, the thalamic reticular nucleus, and the interneurons. So one of the main challenges with training spiking neural networks is that spikes are discrete, they are therefore non-differentiable, and which is why traditional backpropagation algorithms could not be used to train them in the past. Now we have SNN Torch, which is a Python library. It overcomes this challenge by using membrane potential, which is continuous in nature. We have used SNN Torch to build our network. And SNN Torch has already been demonstrated as capable of image classification on the MNIST dataset. Our contribution is that we have presented for the first time a neuromorphic network which is capable of successfully performing image classification. We'll be calling this the thalamus SNN. We've trained on the MNIST and Fashion MNIST datasets with 93.72% and 74.34% accuracy respectively. We've also done a case study on extended MNIST with letters and byclass variants, and that has accuracy of 65.16 and 59.36% respectively. Moving on to the methodology. So this is the leaky integrate and fire neuron. In equation 1, we see when this neuron model emits a spike. So when the membrane potential U at time step t exceeds the threshold value theta, it emits a spike, else it does not. The second equation shows how the membrane potential changes over time. Beta is the membrane potential decay rate, U is the membrane potential, W is the rate of the connection, X is the input at time step t, and the last term is the reset mechanism. So when there's a spike, the threshold is subtracted, else it's not. In the bottom right, we see a raster plot. X-axis is time and y-axis is the neuron index. Here is just one neuron. So the vertical lines represent the presence of a spike and the spaces between them represent the absence of a spike. The digital representation is shown below, a 1 at every time step where there's a vertical line and a 0 at all the other time steps. To begin with our implementation, we refer to the SNN Torch tutorials where they implemented a simple 3-layer architecture. The number of input neurons is 784 because in the MNIST dataset the number of pixels in an image is 784. The output layer has 10 neurons because there are 10 classification categories and there are 1000 hidden layer neurons. Here the connectivity maps are referred to as FCs because by default in SNN Torch the connections are fully connected. And this is a simple architecture that was implemented in the tutorials, and our aim was to test a more brain-inspired neuromorphic network. So we modeled the lateral geniculate nucleus. This is our network architecture. It has the same neuron populations that we discussed earlier, TCR, ERN and IN. And the FCs you'll notice are either red or blue. Red is for an excitatory connection which encourages the firing of other neurons, and blue is an inhibitory connection which suppresses the firing of other neurons. FCN is neither red nor blue because it's not a biological connection. FC9 and FC10 are the noise input to the network, which is Poisson noise, which is introduced to the TCR and IM populations to test our network's robustness to noise. In the table, we see the number of neurons in each neuron population. These have been scaled down by 60% to increase sparsity in our network. And the ratio of the number of neurons in each population was taken from the paper, A Spiking Neural Network Model of the Lateral Geniculate Nucleus on the Spinnaker Machine by Basabdatta Sen Bhattacharya et al.
So input to the TCR and IM population. So the publicly available and most heavily used dataset is the MNIST dataset, which consists of 70,000 images of handwritten digits and has the standard 10 classes. Fashion MNIST dataset, which serves as a drop-in replacement for MNIST with 10 clothing categories in place of digits. Encouraged by our results for MNIST and Fashion MNIST, we decided to try Extended MNIST, which is a more complex dataset. And it has around 6 variants, out of which we have used the MNIST by-class and MNIST letters dataset. By-class consists of 26 classes with uppercase letters, digits and lowercase letters, while the letters dataset consists of 26 classes with case-insensitive letters. So unlike MNIST and Fashion MNIST, our intent was not to train extensively on eMNIST. Instead, we treated it as a case study and we only used the 2 variants to test our network's performance. And along with, other than the image datasets, we have also included noise as input to the TCR and IM population. Now we will be presenting our results. So we have used the default threshold value of the LIF, which is 1. The membrane potential beta decay rate, which is the decay rate, it was set heuristically, 0.9 for TCR, TRN and output population and 0.95 for IN population. The beta decay rate and the threshold value were both made learnable. The FC connections were drawn, the FC connections were generated from using the NN.linear, which draws connection weight values from a uniform distribution ranging from negative root of 1 by k to positive root of 1 by k, where k is the number of neurons in the projecting layer. We have used a 70-30 train test split on all of our data sets, and the training was done for 5 epochs of 380 iterations each. And each iteration and within each iteration, a batch of 128 images was trained for 25 milliseconds. A loss was computed at the end of each epoch, and using the inbuilt cross entropy loss function. And this loss was then back propagated to the network using the inbuilt Adam optimizer with the learning rate parameter eta as 5 into 10⁻⁻⁴. The accuracy of the test data set was calculated as the total number of correctly classified images, which is 5, divided by the total number of classified images. So, which is phi plus web beta. We ran 4 trials, and each trial consisted of 5 epochs for all the data sets, so that we could get the average accuracy. So we can see here that the accuracy barely drops for MNIST as well as fashion MNIST, even with the addition of noise. We saw a similar trend for the accuracy and loss of the EMNIST dataset. This here is a stem plot which depicts the membrane potential for the MNIST dataset. It shows how the membrane potential is used for image classification in a network. The x-axis shows the neuron indices and the y-axis shows the membrane potential value. Let's take a look at image 5, which is the Hamilton digit 0, which is represented by a purple triangle in the graph. We see that it has the highest membrane potential for the zeroth neuron, which matches the predicted. So the predicted class actually matches the target class. Similarly, this is a stem plot for the fashion MNIST dataset, and in the same manner, the neuron with the highest membrane potential matches the target class of that particular image. Similarly, for EMNIST dataset. In conclusion, our SNN Thalamus built on SNN Torch is capable of providing high accuracy in image classification even with the presence of input noise. Our results demonstrate that even small-scale neuromorphic architecture with sparse connectivity can perform similar to conventional neural networks. Performance comparison with a 3-layer SNN gave a higher accuracy for all the tested dataset except MNIST, where our network performed better. Future work could involve varying the parameters, adding noise as input to the image, and also increasing the sparsity. So this work was undertaken as a final year undergraduate thesis under the supervision of Dr. Basabdatta Sen Bhattacharya, and to whom we are grateful for her mentorship throughout the course of this work. We are also thankful for the Department of CSIS of BITS Pilani Goa Campus. This is a picture of all of our co-authors. So pictured from left to right is Alicia Gonsalves. This is Karen Pereira. That is Professor Basabdatta Sen Bhattacharya. That one is me, and our other co-author Arya. Our code will be made available soon after the conference on the GitHub repository under WinLab organization. So thank you for attending the